Our topic for today is beginning to talk about the process of translation. So this is the translation of the genetic information that's in the mRNA into a protein that's actually going to carry out some specific function in the cell. but you can actually get compensatory frame shifts where an insertion and a deletion very near each other combine and cancel out. And the possibility of these compensatory frame shifts is actually some of the earliest evidence that the genetic code was a triplet code in this way. That if you have an insertion here and a deletion in the next codon, it will change these two affected codons, but because you move +1 and then -1, the reading frame is restored after this, and you're much more likely, at least, to get an intact and functional protein. So the sort of deciphering the genetic code was sort of a major question in molecular biology. And I'd like to talk a little bit about the ways, sort of the logic and the reasoning that people used to understand what the genetic code was. So at the time that this was done, with very limited ability to synthesize specific nucleic acid sequences. So the easiest thing to do was use chemical or enzymatic reactions to create completely repetitive homopolymers, where, for instance, you have just uridine. So you take a bunch of, for instance, UTP, and you can incubate it with an enzyme that's simply going to synthesize an RNA that's all U's. And you can take that RNA that is all U and put it into a translation, put it into a cytoplasmic extract that's capable of translation, and ask which protein sequence is encoded by an RNA that's all U. Because it's all uridine, every codon on that message is going to be the same. It's going to be the UUU codon. And it turns out that this message templates the synthesis of a polyphenylalanine protein. And for that, you can conclude that a UUU codon is translated into phenylalanine. Likewise, you can create this homopolymer of all C or all A, and see that those codons that a CCC and an AAA encode just proline or just lysine. And so this sort of reasoning can get you 3 codons, but it can't get you that far. And in fact, it can't even get you the all-G polymer. It turns out that an RNA consisting entirely of Gs folds up into this weird structure that can't be translated. So in order to go beyond these 3 homopolymers, there were a couple of strategies. The first thing that people did was to take a mixture of, say, mostly A and a little bit of C, and make that that polymer. And so what you get from that is you get mostly AAA codons. We already determined that those encode lysine. But you also get some codons with Cs scattered through them. And it turns out that codons with 2 A's and 1 C, those encode amino acids like asparagine, threonine, and glutamine. And so when you translate this, you find that you actually get lots of lysine, a little bit of some of these other amino acids, and in fact you can get, for instance, a little bit of histidine, which is encoded by a codon that actually has 2 C's and 1 A in it. So using these sorts of random polymers, you can make some further inferences about the nature of the genetic code, but because it's totally random, it actually turns out to be very hard to figure out, when you have threonine, is threonine encoded by ACA or CCA? You can tell that it's encoded by a codon that has 1 C and 2 A's, but you can't tell which one that is. And so there's a major advance by a scientist named Karana, who figured out a way to synthesize a repetitive template with polynucleotides. So he figured out a way to make essentially an AC monomer and then polymerize that. So now instead of this sort of random sequence, you have a repeating sequence of ACA, CAC. And it turns out that this thing encodes repeats of threonine and histidine, and this lets you essentially infer, based on the information that you already have here, that a threonine is encoded by a codon with 2 A's and 1 C, and a histidine is encoded by a codon with 2 C's and 1 A, based on sort of the statistics of how much of each of those amino acids you get from the random polymer. When you have this polymer, you can say, okay, the codons you're going to get are ACA and CAC. So the 1C2A codon is this threonine, the 2C1A codon must be histidine. And so these sorts of experiments with these random polymers and these designed repetitive polymers eventually allowed people to work out the entire genetic code. And this is this table here. I don't expect anyone to memorize the entire codon table, but I think there are a few specific features that are important to remember, a few specific codons. And so those are, first of all, that the AUG codon is an initiator or start codon. And this is always a methionine. So this encodes the amino acid methionine either in the middle of a protein or at the beginning. And so every protein essentially begins with a methionine. And there are a lot of other factors that we'll talk about next time about how the cell picks which AUG codon to start at. I mentioned that picking the AUG codon sets the reading frame for the whole message. So picking that AUG codon, the right AUG codon, is extremely important. And there are also 3 stop codons. These don't correspond to any amino acid in the cell, and in fact, they signal the end of the protein. When the ribosome hits a stop codon, there's no tRNA that can come in, and translation is terminated. So the genetic code is degenerate, which means that there are more codon triplets than there are amino acids to encode. There are, in fact, 64 triplet codons. 3 of them are stop codons. That leaves 61 codons for only 20 amino acids. And so what that means is there are many synonymous encodings for a given protein sequence. So a few amino acids, like methionine and tryptophan, have only 1 codon. Most of them have at least 2, and in fact, a few of them have as many as 6 different codons that can encode the same amino acid. So there's a lot of ability, first of all, to make substitutions in a protein sequence that don't change the, substitutions in a nucleotide sequence that don't change the protein that that sequence encodes. The degeneracy in the genetic code is not random. So if you look at this, what you can see is that often there are pairs of synonymous codons that differ specifically at the 3rd nucleotide of the triplet codon, and usually the pattern is that the 2 places where that's a pyrimidine will encode 1 amino acid, and the 2 places where it's a purine will encode a different amino acid. And then in some other cases, we see quartets of synonymous codons where any 3rd base nucleotide is allowed. This observation led to the idea that the genetic code allowed what's called wobble base pairing. So that is a non-Watson-Crick base pairing allowed specifically at the 3rd nucleotide position of the codon to allow a single tRNA to decode 2 different codons. So to remind you, the tRNA base pairs with a codon in order to recognize it, and the specific part of the tRNA that base pairs with the codon is called the anticodon. Because this base pairing, like all nucleotide base pairings, is anti-parallel, that means that the 3rd nucleotide of the codon in the mRNA corresponds to the 1st nucleotide of the anticodon on the tRNA. And at that position, the 3rd codon, 1st anticodon nucleotide, GU base pairs are actually allowed in this. So to remind you that a GU base pair is actually relatively favorable, so it's sort of drawn here that the guanine can present an appropriate hydrogen bond acceptor and a hydrogen bond donor to the U. And in fact, the reason that these don't show up in DNA and in RNA replication and transcription, respectively, is that the polymerase active site specifically disallows this conformation of the T or the U that is needed to make this base pair. So it's relatively energetically favorable. It's forbidden by the polymerase active site. It's also forbidden by the ribosome in the 1st and 2nd codon position, but it's allowed in the 3rd codon position. And this is called wobble base pairing. And it's sort of diagrammed here where a single Lucille tRNA can pair a G in the anticodon with either a C or a U in the codon in the mRNA. And so the idea is an anticodon G can pair either to a C or a U in the codon. And likewise, the anticodon U can, of course, have a Watson-Crick pair to an A, or have a wobble pair between the U and the G. And this pattern of pairing with anticodon G and anticodon U explains the structure of the genetic code, where we see that the redundancy occurs when you have, either this AG in the codon corresponding to a U anticodon, or a CU codon corresponding to a G anticodon. So there are actually some cases where, even though you could have this sort of wobble decoding, some organisms have a tRNA that directly decodes, which mean Watson-Crick base pairs, with both a C or a U here, but this wobble pairing gives you at least the potential to recognize all codons, all of these codon pairs that are synonymous with the third site, with a single tRNA for each pair. There's actually an additional kind of wobble pairing that emerges in a few cases, and this relies on the nucleotide inosine. So inosine is the product of deamination of adenosine. And in a few tRNA codons, an adenosine that's actually encoded in the tRNA gene is modified to an inosine by an enzyme. And that allows the creation of an inosine wobble pair. So inosine has a fairly canonical Watson-Crick pairing with C, but it can also form weak base pairs both with U and A. And so on a codon that's modified, you have an inosine in the first anticodon position, which is going to recognize the third codon position. A single tRNA can actually recognize a U, A, or a C. And this is sort of a particular feature, the kind of base pairing that's allowed at this third codon position in the ribosome. So the transfer RNA is diagrammed as sort of the secondary structure is shown here. And the anticodon is sort of in the middle of it. So this structure runs from the 5' end here. It folds up into these stem loops here, where it base pairs extensively with itself. And then right at the 3' end is the place where the amino acid is actually attached. Yeah?
So is conversion of adenosine to inosine unique only to the translation process, or can it be also involved in transcription?
Okay, so the question is adenosine to inosine conversion specific to translation. So I believe there are certain cases where mRNAs can be edited after they're transcribed, but in general, you're not going to see inosine base pairing during replication or transcription. It really is a phenomenon that comes up in translation, except in the case of, say, DNA damage, where the A might be converted to an I. That would be the only case that's something that would normally be corrected. So the transfer RNA secondary structure looks like this, with base pairing between the beginning and the end of the tRNA, as well as these two arms. There are, in addition to the possibility of things like inosine in the anticodon, there are a wide variety of base modifications here. The places highlighted in pink here are residues that are totally conserved in all tRNAs. And those include this modified pseudouridine, as well as some other very unusual nucleotides. And in fact, the anticodon itself can be, those bases can be extensively modified by methylation in a way that seems to really tune the recognition of the anticodon by the codon. So there are sort of two constraints on the tRNA. All tRNAs have to work with the ribosome. They all have to come into the ribosome, base pair with the anticodon, and bring in the amino acid. But they each have to be different enough to sort of bring in base pair with a different codon and bring in a different amino acid. And so that's really sort of the line that they have to walk, and that probably explains in part why the tRNAs are so extensively modified. They're more highly modified than the rRNA, and certainly than any coding mRNA in the cell. So the three-dimensional structure of a tRNA has this sort of L shape. So it folds up. This is sort of showing the diagram here, sort of a space-filling model. And the two key features are the anticodon loop down here, and the amino acid arm up here where the amino acid is actually attached. So you can see that this stem loop here corresponds to this region here. This arm here is in blue here, this one here in orange, and so they sort of fold down, and then the amino acid arm sticks out here, so it's sort of L-shaped in 3 dimensions. And the amino acid is attached by an ester linkage to the 3' OH at the end of the transfer RNA. And so this is a high-energy linkage that actually provides the energy for peptide bond formation. Once the amino acid is charged onto the transfer RNA, it's actually a downhill reaction to form that peptide bond. And so the hydrolysis of ATP to AMP is used to drive the charging of the amino acid onto the tRNA. So this here is the last base of the tRNA, which is always an adenine and adenosine here, with the amino acid linked onto the 3' OH. And the enzyme that's responsible for doing this is called an aminoacyl tRNA synthetase. So it catalyzes the reaction of an amino acid with the appropriate tRNA and ATP to make this aminoacyl tRNA AMP and pyrophosphate. So here's a diagram so the blue space filling is one of these enzymes, and you can see that it's interacting with the anticodon loop as well as some other parts of the tRNA in order to recognize that it's charging the right tRNA. So it's very important because the synthetase charging the right amino acid on the right tRNA is what actually determines the genetic code. The ribosome is a pretty generic protein synthesis machine, and if you give it the wrong amino acid on a tRNA, it will happily incorporate that wrong amino acid. So there's one synthetase per amino acid, and it recognizes features of the tRNA, including the anticodon loop, but not limited to the anticodon loop, in order to charge the right tRNA with the right amino acid. So there are actually 2 classes of these synthetase enzymes that catalyze the reaction in 2 slightly different ways. And the difference between them is something that we can see both by the structure of these enzymes. The crystal structure has been solved, also by the sequence similarities, and by the mechanism of these enzymes. So in all cases, the first thing that happens is the amino acid, the carboxylic acid is a nucleophile that attacks the alpha phosphate of the ATP. And that creates this high energy aminoacyl adenylate here, where the amino acid is charged onto the phosphate. And then in class 1 synthetases, the first thing that happens is actually this 2' OH is a nucleophile that attacks this linkage to actually attach the 2' OH to the amino acid. In the case of a class 2 amino acid, the 3' OH is a nucleophile. And so this leaves you, this nucleophilic attack is going to actually leave you with a properly charged tRNA where the 3' OH of the last A is connected to the amino acid. In the case of a class 1 synthetase, there's an additional isoenergetic step that has to happen, and it's actually pretty simple. Just the 3' OH takes the amino acid off of the 2' OH, and ultimately the final product always looks like this. There's just this extra step here that goes on in these class 1 enzymes. So I mentioned that the genetic code is really determined by the tRNA and the synthetase, that whatever amino acid the synthetase puts onto the tRNA will be used by the ribosome, and whatever anticodon is present on the tRNA will recognize an mRNA codon. The ribosome is a pretty sort of generic machine for doing this. And so one problem that comes up is this, and the answer, the question is, can you tell these 2 branched chain amino acids apart? The answer is yes, you can, but it's actually kind of tricky. It's hard for the synthetase too. So this is isoleucine and this is valine. There's a mnemonic you can use to recognize it, because valine is like this little V because it's symmetric, or isoleucine is like an L here. But it's hard for us and it's hard for the synthetase, and so the synthetase can actually achieve only about a 200-fold preference for isoleucine over valine. And there's actually a second proofreading active site on the tRNA synthetase. This is particularly important for these branched chain amino acids that have relatively subtle differences. And this is capable of hydrolyzing valyl tRNA onto the isoleucine tRNA. So when you put a valine onto an isoleucine tRNA, which you do about 1 out of every 200 times, that thing is a substrate to fit into a proofreading second active site on the isoleucine tRNA synthetase. But the correct attachment of isoleucine onto that tRNA, because of this extra methyl group here, won't actually fit into the active site. And so that gives you an extra about 10 to 15-fold specificity, meaning that at least 90% of the time this proofreading activity catches the mischarging and hydrolyzes that bond and essentially just tries again to get the right isoleucine onto it. And so this is sort of similar to the proofreading activity we see in, for instance, DNA polymerase, where the polymerase can achieve only a certain amount of specificity for adding the correct nucleotide. And as a strategy to improve its accuracy beyond that, it can detect and hydrolyze an incorrect nucleotide much of the time. In the same way, tRNA synthetase can hydrolyze the wrong amino acid off of the tRNA much of the time. The other side of this equation is that the synthetase has to recognize the correct tRNA. And there are a number of features of the tRNA besides the anticodon loop itself that are recognized, and these are shown with some of these arrows here and sort of depicted here as well in orange and blue. Unsurprisingly, a lot of the features that the synthetase uses to recognize the tRNA, in addition to the anticodon itself, are ones that are going to lie on this surface. So this is where the tRNA interacts with the synthetase, and of course many specificity determinants are going to fall right near the amino acid arm, which is where the tRNA synthetase is actually doing its chemistry. And so this is also very important because the tRNA is larger and there are more ways that it can differ. It's not as challenging for the synthetase to be extremely specific for its cognate tRNA, but it does recognize features outside the anticodon loop itself. So once the tRNA is charged, the chemistry of peptide bond formation is fairly straightforward. It's a nucleophilic attack. So this is the growing peptide, and the growing peptide is going to be linked to a tRNA here. So this is the 3' terminal adenine of one tRNA with the growing peptide linked to it through this ester here. And then an amino acid will come in, and it will also have an ester linkage to a tRNA and the 3' terminal adenine. There'll be a nucleophilic attack by the amine of the amino acid. So the linkage to the nucleotide is with the carboxyl. And so the amine is free to perform this nucleophilic attack on this carbon and ending up kicking it off of this tRNA, leaving behind a free 3' OH here, where this linkage from the 3' now at OH, and the peptide bond has been formed here between this carbon and this nitrogen on the amine. And of course, the product of this peptide, now has its C terminus still linked to a 3' OH. And in fact, this whole process can repeat again with a new amino acid coming in using its amine to attach here. The growing protein always remains esterified, held onto a tRNA as it's growing. And so the setting for all of this is the ribosome. So the ribosome is a large structure. It's about 2/3 RNA and 1/3 protein. And there are two subunits, a large and a small subunit, and they're named based on their sedimentation coefficient. So this is one of the early methods used to separate ribosomes. Is the fact that it's very large and very dense, because they contain a lot of RNA with a lot of phosphate in it. And so they'll sediment during ultracentrifugation, and the speed with which they sediment was used to name them. So the assembled bacterial ribosome is 70S, and the individual components are 50 and 30S. The eukaryotic ribosome is slightly larger, so the subunits are 60 and 40S, and come together into an 80S ribosome. So the ribosome, each of the subunits has a single major structural RNA. It's named for its sedimentation. And so in bacteria, it's a 16S and a 23S rRNA in the large subunit respectively. In eukaryotic ribosome, it's 18S and 28S. There are also a couple of other smaller RNAs in the ribosome. All ribosomes have a 5S rRNA, and eukaryotic ribosome also a 5.8S RNA, so those are fairly small. The RNA, the larger RNAs are almost 5 kb long. So recently, we've actually gotten high-resolution crystal structures of both the prokaryotic and more recently the eukaryotic ribosomes. So you have a much clearer sense of exactly what these look like. And so here is sort of shown now the large subunit is on the right and the small subunit is on the left. And this structure doesn't actually have an mRNA in it, but if there was an mRNA, it would sort of thread through here like this. And the tRNA sort of bridge between the two subunits. Since the mRNA sort of wrapping through this cleft in the small subunit, the tRNA and the anticodon loops are all down here in the small subunit. It's said to contain the encoding center of the ribosome, where the tRNAs are base pairing to the mRNA. In the large subunit is the peptidyl transfer center, where the peptide bond is actually formed. The tRNAs are formed in this L shape here. You can sort of see through here. One thing to note, so the gray and the sort of the cyan here are the RNA. You can see it's all these backbone sheets with these base pairings across it. And all these purple coils, these are proteins. I think that looking at this sort of gives you the sense of the extent to which the ribosome is really composed of structural RNA that's holding the whole thing together. There are 3 tRNA binding sites on the ribosome. It's called the A, P, and E sites. And so in red here is the aminoacyl tRNA site. This is where a new tRNA comes into the ribosome. In the middle is the P site, or the peptidyl tRNA site. So this is a tRNA that's linked to the growing peptide. And finally, there's an exit site. And the steps of the translation cycle start first with tRNA recruitment. So the ribosome has to bring in the correct tRNA and ensure that it pairs correctly with the codon that's going to be decoded. Then, once it's satisfied that it has the right tRNA, there's a peptide bond that's formed here, and the growing peptide is transferred onto this tRNA. And finally, it has to translocate exactly 3 nucleotides to shift this tRNA into the P site, kick out this tRNA, and undergo a new round of tRNA recruitment. So tRNA recruitment is facilitated and proofread by a protein called EFTU in bacteria and EF1 alpha in eukaryotes. So EFTU binds only to charged tRNAs and not to uncharged tRNAs. It binds to all tRNAs with pretty similar affinity, and it delivers those charged tRNAs to the ribosome. And here, this is the tRNA is now shown sort of flipped around. Here's the anticodon. Here's the amino acid. The protein, I think the GTP is buried like right there or something. It binds GTP, and the hydrolysis of that GTP serves as a mechanism to ensure proper base pairing in a couple of different ways. So EFTU, you can see, is holding on to the top of this tRNA. And when it delivers it to the ribosome, the tRNA doesn't move all the way in to the active site. And in fact, EFTU needs to hydrolyze GTP and then release the tRNA in order to have a successful tRNA recruitment. And that process, because the tRNA changes its shape as it moves into the ribosome, is always sometimes called accommodation, because the shape of the tRNA is accommodating to the tRNA binding site on the ribosome. And so this sort of multi-step process is shown here, that the tRNA linked to EFTU with GTP comes in, the activation of the GTPase is dependent on conformational changes that only happen when you have correct base pairing of the tRNA to the tRNA anticodon to the mRNA. And then even after GTP hydrolysis, there's an additional delay before EFT-GDP releases. And this provides an additional time for incorrectly paired tRNAs to dissociate. It's called kinetic proofreading. So the cell is ensuring that in addition to recognizing correct codon-anticodon interactions with the sort of the binding energy of proper base pairing, it's also essentially using the energy of GTP hydrolysis to create additional time and thereby additional specificity for the correct base pairing. There's sort of this trade-off between energy and specificity, and that the GTP hydrolysis is a way of putting additional energy into ensuring that only the correct base pairing happens. Once the tRNA is accommodated into the ribosome, the EFTU needs to be recycled, and so there's a factor called EFTS in bacteria, or EF beta gamma in eukaryotes that comes in and exchanges the GDP for a new GTP. And once in the GTP-bound state, this protein can go find a new charged tRNA, participate in a new round. And the ribosome samples many, many tRNAs at each active site. The tRNA that's coming in and sampling the active site is, every tRNA comes in and samples that active site, and only one of 30 or 40 tRNAs is actually paired correctly. So there's actually quite a bit of this sampling, or this sampling and tRNA coming out at this stage, or this tRNA rejection here, before you finally get accommodation, the acceptance of a proper tRNA codon-anticodon pairing. And then once that happens, this tRNA moves into the A site into a configuration where it's capable of forming the peptide bond. And so we've already talked about the basic chemistry of peptide bond formation. And I think the interesting observation of the way this happens on the ribosome is the ribosome is a ribozyme, which means that, and this is sort of some recent and very sort of high-resolution information about this. It's not something that you need to memorize in detail, but the important point is that the catalytic site consists of 2' OHs, backbone phosphates, and amine groups from bases of the rRNA, and in some cases the tRNA, and there's essentially no involvement of protein here. So the ribosome itself is a catalytic RNA. So after the peptide bond, the new peptide bond is formed here, the next step is the ribosome has to undergo this translocation step. So this translocation is catalyzed by another GTP binding protein called EFG in prokaryotes and EEF2 in eukaryotes. And the sort of the net process here, this is now shown going right to left. So after the peptide bond has been formed, you need to step the EFG binds, and you need to step the tRNA in the A site into the P site. The deacylated P site tRNA needs to move over into the E site. And this is coupled, this is made to go into one direction by the hydrolysis of the second GTP by EFG. So there are two GTP hydrolysis events that occur in addition to probably many other sort of futile GTP hydrolyses that occur during amino acid, during tRNA selection. And this process of translocation occurs through a rotation of the two subunits. So from this state, the ribosome rotates the large against the small subunit. And what this means is that as this rotation happens, the way it's done here, the tops of these tRNAs, the amino acid stems, move from their positions in the peptide transfer center in the P and the A sites into the E and the P sites, while remaining paired like this on the small subunit. So this whole thing is rotated. And then in conjunction with GTP hydrolysis, this moves back, and it slides 3 bases, and the tRNAs always remain paired to the mRNA, and this ensures that you keep the same reading frame, that this base pairing here moves to this base pairing here, and ensures that the codon here moves exactly 3 bases into the codon position for the P site tRNA. And this process happens with pretty high fidelity most of the time. So the ribosome, as I said, can translate a protein that is over 10,000 amino acids long and not lose the reading frame most of the time. There are a few specific cases, though, where the ribosome does change the reading frame as a result of some features of the mRNA sequence. So this is called programmed ribosomal frame shifting. And what can happen is a combination of 2 different effects can happen at the same time. First of all, you have a highly structured region of RNA that is going to block translocation. So of course the mRNA threads through the ribosome as a single-stranded RNA, and so if it has extensive base pairing to itself, that process is inhibited. And also the reading frame of the ribosome is maintained by base pairing between the tRNAs and the mRNA. And so in these, when you have the combination of an RNA structure that blocks ribosome translocation, something called a slippery site, where the A and P site tRNAs could repair one nucleotide over and still have acceptable base pairings. These 2 things together can promote the ribosome to shift into a different reading frame. And probably the most notable example of this occurs during the translation of the retroviral and retrotransposon reverse transcriptase. So here down here is shown HIV is sort of an example of these proteins that have, these genomes that have a GAG reading frame that encodes a number of structural proteins, as well as a Pol reading frame that encodes the reverse transcriptase itself. And in HIV and in many retrotransposons, actually the GAG reading frame terminates right at a position where there's a frame shift somewhere between 5 and 10% of the time into a separate reading frame that produces a reverse transcriptase, which is needed at sort of lower concentrations than the structural proteins. And so there are a few cases of cellular genes that have a similar mechanism where the reading frame can change at a programmed frame shifting site to produce 2 different versions of the same protein from a single mRNA sequence. But this is definitely the exception and not the rule. It occurs only when you have the coincidence of this very strong RNA structure with a specific site, a specific slippery site that will allow the tRNA bases to repair. And in some sense, sort of the importance of this tRNA repairing is the exception that proves the rule that base pairing between the tRNAs and the mRNA is what maintains the reading frame, as well as what gives you sort of the specificity for specific codons. And sort of putting together this whole translation elongation cycle here, the process to add a new amino acid is the recruitment of the tRNA and its eventual accommodation into the ribosome based on the proper base pairing between the codon and the anticodon that allows wobble base pairing at the 3rd position, but not at the 1st or 2nd. When this accommodation is complete, peptidyl transfer can occur, and an EFG is able, once peptide bond has been formed, EFG can translocate the ribosome 1 codon, 3 nucleotides down based on this stable interaction between the tRNAs and the mRNA as the subunits rotate against each other and then rotate back. And so, getting towards the end, I'd like to ask a question about this process. The question is that 20 20 major amino acids for naturally in proteins, but people recently been interested in incorporating some non-standard amino acids. And the question is, what strategy would be, do you think, the best to engineer a bacterium to incorporate a new amino acid?
This is going to be my downfall. Do you even know what EFT is? Let's see, change. That's too hard. You can't change A. I think you can change the ribosomes. Maybe it's A.
What is charged in the amino acid? What is charged? I put B. What about E? I don't know. E is technically. Or E. So is B. B?
You'll pick one.
What's EF? Oh,
What?
fuck, 3 seconds.
It's EF. Are you sure? Is it B?
I picked A.
Is it B? Oh, so hard. Oh my god, I don't know if I did it. I don't know if I did it in time.
Yes! Wait, Tim, did you get it?
I don't know if I did it in time.
The answer
I think I did.
Why is it?
You're gonna hear me scream and you're like,
tRNA synthetase
I put
oh my god.
need to
I don't know
change
if I did it in time. I
amino
didn't
acids.
look
Remember
down.
that the tRNA synthetase and the tRNA are the things that really determine the genetic code. So the ribosome is a generic machine that will form peptide bonds with anything that's charged under a tRNA, including natural amino acids like phenylalanine and tyrosine, as well as this and even much weirder amino acids that people have tried. And all of those things, far beyond the 20 natural amino acids, can make proteins. And the rest of the machinery, both the ribosome as well as elongation factor TU, will accept any tRNA amino acid charging. And so it's really the synthetases of the tRNA that determine the genetic code. So I think actually we will stop here for today and we'll begin with tRNA.